Hello everyone, my next topic for this educational series is on omics. You probably heard this term lots of times during your study. So what are omics, what they mean, and how they work helping clinicians? If we make a journey from genotype towards phenotype, we encounter different molecules including transcriptomes and proteomes, which are the two key biomolecules family having great impact on recent molecular biology and immunology area. And today I will be focused on these two molecules, transcriptome and the proteomics. The first human genome project was an international scientific research project with the goal of shaping the base pairs that make up our DNA molecule and to identify, map, and sequencing all the genes of our genomes from both physical and functional standpoint, which we name it as a book of life. And this book took about 2.7 billion and almost 15 years to be completed. Okay, so what is omics? The word omics refers to a field of a study of biological science that ends with omics, such as genomics, transcriptomics, proteomics, metabolomics, epigenomics, and so on. Omics aims at the collective characterization and qualification of pools of biological molecules that translate into the structure, function, and dynamics of an organism. So how to integrate omics into the central dogma? As we know, the central dogma starts with the genome, which is a complete DNA content of a live cell. And then through the transcription, we have got transcriptome, which is a complete set of transcripts and relative levels of expression in a particular cell or tissue under defined condition at a given time. Following the proteomics, which is complete collection of the proteins and their relative levels in each cell. So as I mentioned, for this talk, I'm going mainly focus on the transcriptomics as well as the proteomics. So before I go forward, I just want to make sure to clarify genomics from transcriptomics. So genomics provides an overview of the complete sets of the genetic instructions provided by the DNA, whereas the transcriptome investigates the gene expression pattern. And this in transcriptome term was first proposed by a scientist named Charles Offrey in 1996. So in a simple way, I can just mention that transcriptomics is like a bridge connecting genomics to the proteomics. Okay, so have you ever had to transcribe something? Maybe someone left a message on your voicemail and you had to write it down on a paper. Or maybe you took notes in a class, then you rewrote them neatly to help you to review them. As these examples show, transcription is a process in which information is rewritten. This is something that we do in our everyday life, and it's also something our cells must do, but in a more specialized way. So a study of the transcriptomes allows us to understand the expression of genome at the transcription level and provide information on gene structure, regulation of gene expression, gene product function, as well as genome dynamics. So what is really in our DNA? For many years, it has been thought much of our genome has no apparent purpose. These are the DNA sequences that they don't encode for a protein, and they were known as a junk DNA or broken gene. Why we not look like a chimpanzee, or despite we are sharing more than 90% of the same DNA, or why some people are taller or they have got higher blood pressure, or why some people they find it more difficult to lose weight. To answer all of these questions lies on genetic materials previously known as a junk DNA, which we all know they work in so many important ways and they contain most of the information that can turn on or off the real genes. So we are in a golden age of understanding non-coding DNA and non-coding RNA. So I think it's a good time to most logical explanation for now to say that the junk DNA might not be so useless after all. Okay, so I'm just going to explain a little bit about coding and non-coding DNA. What is the coding DNA? The name for coding DNA is protein coding DNA. They are DNA that accounts only for 1% of the human genome. They are composed of exons. They encode for proteins and undergoes transcription to synthesize messenger RNAs. The proteins encoded by coding DNA have a structural, functional, and regulatory importance in all of the life cells. We have got non-protein coding DNA, which they account for more than 99% of the human genome. They are composed of regulatory elements, non-coding RNA genes, introns, pseudogenes, repeating sequences, as well as telomeres. These non-protein coding DNA does not encode for proteins, and they undergoes transcription to synthesize RNAs like transfer RNA, ribosomal RNA, and other regulatory RNAs. And these non-protein coding DNA, they are important for controlling genes activity. Okay, so when it comes to the non-coding sequences, they can be grouped in different groups. So one of them is a structural gene, which they are not transcribing. As an example, I can just mention centromeres, which are part of chromosome attached to the spindle fibers during the cell division, and it allows the chromosomes to be pulled to the opposite ends of the cell. And we have got telomere that help to maintain a structure of the chromosomes in eukaryotes, which I'm going to explain more about this telomere later today. Okay, so the second group of these non-coding DNA, they are introns, they are transcribing, but RNA is removed through splicing before translation. And the third group are functional DNA that they are transcribed but not translating to any protein. And I can just give an example like transfer RNA and ribosomal RNA, which are the two key molecules during the translation of the transcriptome to a protein. So when it comes to the non-coding RNA, we have got a small non-coding RNA, which the size is around 20 to 24 nucleotides or base pair. And we have got long non-coding RNA, which the size is usually more than 200 nucleotides. Non-coding RNAs refers, as I mentioned, refers to all of the RNAs that are not translated to functional protein, but they act as cellular regulators. Abundant and functionally important types of non-coding RNA includes any type of RNA which is not messenger RNA, and I can just give an example like transfer RNA and ribosomal RNA, as well as molecules like miRNA, which is microRNA, and they are silencing and post-transcriptional regulation of the gene expression. We have got siRNA, which is a small interfering or silencing non-coding RNA, which they can cause gene silencing through repression of the translation. The next one is piRNA or PV interacting RNA, which involves in epigenetic. We have got a small nuclear RNA, which assists processing of pre-messenger RNA into the nucleus, and also it can help in the regulation of transcription factors and maintaining the telomeres. The next example for non-coding RNA is a small nucleolar RNA, which can guide chemical modifications of other RNA, mainly ribosomal RNA, transfer RNA, and a small nuclear RNA. And finally, we have got extracellular RNA that there is no biological function fully understood for this xRNA, but they have been proposed to play a role in a variety of the biological process, including intracellular communication and cell regulation. As I mentioned, based on the size of the non-coding RNA, we have got long non-coding RNA as well, which are the key players in many cellular functions, such as splicing, gene regulation, chromosome structure, and hormone-like activity. Long non-coding RNA, I can just give an example. It has been shown that there is a long non-coding RNA X inactive specific transcript with the size of 17 kilobase pair on the X chromosome of the placental mammals, which can act as a major effector of the X inactivation process. This long non-coding RNA can orchestrate the epigenetic transcriptional states of the 2 X chromosomes in female, as well as the 1 single X chromosome in males. So in general, it can prevent them from having twice as many X chromosomes in both gender. So another example of when a non-coding RNA play an important role is, as I mentioned, the telomere. Inside the nucleus, genes are arranged on chromosomes. At the end of the chromosomes are stretches of DNA, which we call them telomeres, and make it possible for cell to divide. Telomeres have been compared with the plastic tips on the shoelaces because they keep chromosomes end from sticking to each other, which would, if it happens, it can destroy the organism's genetic information. So as I mentioned, telomere is the end of the chromosome, and it is made of a repetitive sequence of non-coding gene that can protect the chromosome from damage. It has been shown that every time a cell can divide, the telomeres become shorter. So when we are born, we have got a nucleotide size of around 8,000 nucleotide on our telomere, and during the life, when the cells start to divide, when we age, it can end it up with a nucleotide size of around 1,500. So aging, inflammation, cancer, and oxidative stress can accelerate the process of telomere shortening. How to study the transcriptomes? Here I'm going to give you an examples of technologies to study transcriptomes, which includes DNA microarray, which is a hybridization-based technique, and RNA-seq, which is a sequence-based approach. There are a few issues with microarrays, including poor quantification of lowly and highly expressed genes, and needing to know the sequence to start with the technique. And because of these technical issues, transcriptomics transitioned to sequence-based method with current technology named as next-gene sequencing of complementary DNA, which we name it as RNA-seq, and it can pretty much provide an unbiased insight into the all transcripts in a living cell. Okay, so the next step in central dogma and omics is proteomics, which is more complicated than genomics because an organism's genome is more or less constant, whereas the proteomes differs from cell to cell and from time to time. So when it comes to the central dogma, in genomics part, the DNA can tell what is possibly happening. In the second part, which is a transcription or transcriptomics, the RNA can tell us what probably can happen. But at the end, the proteins are the actual molecule that's telling us what is really happening in the cell. Okay, so the proteins are key building blocks of a living organisms with many function and are kind of an expression of organism's genome. To demonstrate the importance of these molecules, protein is originated from a Greek word which is meaning first rank. Our body contains millions of proteins. However, they are not all produced by all the body cells at any given time. The proteins helps to repair and build our body tissues, allows metabolic reactions to take place and coordinate body function, also providing our body with a structural framework, maintaining proper pH and fluid balance. They can act as a hormone or they help in the defenses system, and this list can go on and on. The proteomics focus on the identification, localization, and functional analysis of the protein makeup of the cells. Also, the proteomics can tell us the protein's function, their subcellular location, which could change with the organisms, and the condition faced by their host cells, including age, checkpoints in the cell cycle, and external or internal signaling events. So why the proteome or protein are that complex, and where the proteome complexity comes from? A protein coding gene not just making one protein, it's making an average of 7 or 8 different versions of a protein from the same gene. Actually, after the gene is copied from the DNA, it can mix and match different parts of a gene to make completely new protein. And also different post modification, translational modification system can work in here. These are including phosphorylation, glycosylation, methylation, acetylation, and lipidation. Given the number of the proteins that can be produced by individual organisms, we can bring up some applications for proteomics. So proteomics allows greater understanding of the complexity of life and the process of evolution than the study of the genetic code can do it on its own. So here I'm going to show you some of the applications of the proteomics. It can be involved in lots of different areas in research, including cancer research, nutrition research, diabetes, tumor metastasis, antibody treatments, neurology, and renal disease diagnosis. As in matter of the clinical proteomics, it covers a spectrum of activity from preclinical discovery to applied diagnosis. So clinical proteomics can close the gap from discovery to implementation in patient management. So as an example for clinical proteomics, I can just mention discovery of the disease biomarker, identification of the drug targets, study of the disease pathogens, personalized medicine, as well as host and pathogen interaction field. Based on proteomics approaches, we have got different techniques that I'm just going to mention them briefly in this diagram. So as I mentioned, if the purpose of the study of a protein is to purify it first, so most of the techniques are based on the chromatography techniques. If we want to analyze the purified protein, we can use ELISA, Western blot, and protein microarray. For characterization, most of the techniques is based on the gels as well as the mass spectrometry. And if we want to analyze the structure of the purified protein, we can use X-ray crystallography as well as NMR spectroscopy. And when it comes to the quantification and analysis and bioinformatician, there are lots of different softwares and programs to help us to go through them. So bringing up omics together as a summary, what can transcriptome and transcriptomics tells us? Transcriptome of a cell is a collection of all of the RNA molecules or transcripts present in one cell. Also, transcriptome analyzes, it can reveal the regulation network of biological process and give some guidance in disease diagnosis and clinical therapy. Transcriptomics undoubtedly deserves to be considered as a contemporary game changer in many diseases, including cancer, on both molecular and clinical level. What can proteomics tells us? Proteomics is one of the most significant methodology to comprehend the gene function, although it's much more complex. Proteomics enables the identification of ever-increasing number of the proteins, and integration of proteomics with genomics and transcriptomics can provide some promising diagnostic and therapeutic targets for various disorders. So thanks again for the opportunity to speak today.